Hello friends, my today's talk is on prokaryotic and eukaryotic cells. Pro means first, karyon means nucleus, that is the stage before the formation of eukaryotic cells or the cells without conspicuous nucleus or without a distinct nucleus. U means two, karyon means nucleus, that is cells with two nucleus stands for the eukaryotic cells. A typical example of eukaryotic cell is yeast cells that grow rapidly and are used as experimental organisms. It is common baker's yeast that is used in bakery and also known as brewer's yeast used in preparation of beer. So this is a simplest one. It is Latin name is Saccharomyces cerevisiae. So this is a very typical example. Otherwise also two terms, yeast and molds, are of great importance for a student of microbiology or industrial microbiology or biotechnology. Egg cell is a typical example of an animal cell. This is Xenopus laevis, the cell of the frog, which has got hereditary material to complete itself to convert into an adult frog. 10 million to 100 million living species exist. And what is the major thing that differentiates living cells from non-living material is that they have got the hereditary material. Both living and non-living may grow. The living cells grow and a crystal of chemical may also grow in size. But that is not life. That is not life. Oxidation reduction occurs in the living cells, and while a candle is burning, that is in that also. There may be the waves of water in the ocean, but that does not define the life. The life is that hereditary material is there. Living organisms can produce organisms like themselves, their offsprings like themselves. So this is the major characteristic of life. And what is that hereditary material? That is, all cells store their hereditary information in a linear chemical code, that is DNA. DNA is unbranched polymer chain, deoxyribonucleic acid. Deoxyribonucleic acid, it passes on the hereditary material and is found in all living organisms, prokaryotes as well as eukaryotes. All cells replicate hereditary information, and DNA comprises of smaller units called nucleotides. Each nucleotide consists of a sugar phosphate bond. a sugar bond, ribose sugar, to which the phosphate group is attached, and nitrogenous bases. The nitrogenous bases are adenine, abbreviated or written as A, guanine written as G, cytosine written as C, and thymine written as T. So this makes the hereditary material, the nitrogenous base in the sugar phosphate backbone. All cells transcribe portions of their hereditary information in intermediary form, and that intermediary form is the RNA. Here, it will be worthwhile to tell that there is a classical question whether hen came first or egg came first. The same pattern, it may be asked whether DNA was formed first or RNA was formed first in the origin of life. So it has been proved now that RNA was formed first, and this is the intermediary form. RNA is the intermediary form of the DNA. DNA can lead to the formation of protein, which is an essential feature of all living organisms, all cells, prokaryotic as well as eukaryotic. It may be divided into 3 stages. The first stage is the replication. Replication is the duplication of DNA, and in the second stage is the RNA synthesis, and third is the protein synthesis. So, first of all, DNA to DNA, this is the replication or the duplication, and then from DNA to template of RNA is formed, and which is regarded as the transcription. And from transcription, after transcription comes the RNA synthesizes the protein, and protein synthesis is known as the translation. So these are the 3 stages, that is replication, transcription, and protein synthesis. Different amino acids form a chain and result into formation of a protein. All cells use proteins as catalysts. Protein molecules like DNA and RNA molecules are long unbranched polymers. So some proteins, they work as catalysts. They may be regarded as biological catalysts or biocatalysts that are essential for the activities of living cells. The intermediary form, that is the RNA, the conformation of RNA is very interesting. Nucleotides pairing between different regions of the same RNA polymer chain causes the molecule to adapt distinct shape, and it is visible. It can be seen in the G, C, A, U, G, C on one end, and C, G, U, A, C, G, U, A, U, G on the another end, another side, and in this way complementing each other, and this is how the conformation of RNA takes place. All cells translate RNA into protein in the same manner in the process of translation in prokaryotic as well as eukaryotic cells. So this is a common feature of prokaryotic as well as eukaryotic cells. Fragment of genetic information corresponding to one protein in one gene. It believed that one gene results the formation of one protein, but sometimes more than one gene may be responsible for the formation of a protein, and even some codons, they are not functional. They are regarded as the nonsense codons, and it does not mean anything. Life is a process that requires energy, and without energy life cannot go, and this comes in a series of reactions that go in the prokaryotic as well as the eukaryotic cells. All cells function as biochemical factories dealing with same basic molecular building blocks. So these biochemical factories, they carry on various reactions, and these factories never go on strike. They are very disciplined and carry on different biochemical reactions, and different sort of products are thus formed, and which are harvested by microbiologists and biotechnologists. All cells are enclosed in a plasma membrane across which nutrients and waste materials must pass. Plasma membrane is the characteristic feature of all living organisms. This plasma membrane surrounds the cell cytoplasm, and this plasma membrane might have originated under very harsh conditions, and in such a way that some proteins are also present in the plasma membrane. They may be surface proteins, they may be intrinsic proteins, INTRINS, intrinsic proteins. So the surface proteins and intrinsic proteins, with the help of these proteins, certain substances required as nutrients may pass in, and those that are not required, the waste substances, may go outside. All living cells exist with fewer than 500 genes. A very limited number of genes cells can manage their activities of life. The phospholipid monolayer and phospholipid bilayer of this, they form the membrane. How it might have been that it might have been there under harsh conditions, and even can be today if you place a drop of oil on the surface of, above is oil and in the beaker and below is water. So in the oil part, phospholipid monolayer may be formed, and in the water biophospholipid bilayer may be formed, and in the oil these heads are downward, and the stains, they face towards the oil. Perhaps this is the theory and how the formation of monophosphate and biphosphate lipid layers might have occurred in nature, and this simple experiment demonstrates. A very interesting protein is the ABC transporter protein, also referred as the ABC protein, a large superfamily of transport proteins which use energy of hydrolysis of ATP to transfer peptides. So this ABC transfer proteins, also referred as ABCT proteins, these are very essential ABC transfer proteins. Cells can be powered by a variety of energy sources. The energy sources may be some organic substance, and the cells are regarded as organotrophic. The source of energy may be light. The cell is regarded as phototrophic. Photo means light, trophic means nutrition, and the third category is the lithotrophic. Litho means rock or inorganic material, and trophy means nutrition. So lithotrophic cells are those cells in which rocks are or elements are required for the nutrition. And in the lithotrophic, one of the typical example of the lithotrophic cells is the bacterium Bigiatoa. Bigiatoa lives in sulfurous environments and gets energy by oxidizing hydrogen sulfide H2S. And in such cells, the Bigiatoa, yellow deposits of sulfur inside the cell can be seen. So, you see here in this diagram, yellow colored granules of sulfur that have accumulated in the Bigiatoa because it lives in the sulfurous environment and it obtains its energy from H2S hydrogen sulfide. Some cells fix carbon dioxide and some cells fix nitrogen. Some photosynthetic cells, they fix carbon dioxide, and typical examples of cells that fix up nitrogen are Rhizobium. Rhizobium is a symbiotic bacterium, and another example of free living nitrogen fixer is the Azotobacter. At the same time, some blue-green algae as Anabina and Nostoc, they are also capable of fixing up atmospheric nitrogen, and some actinomycetes, they also be responsible for fixation of nitrogen. Greatest biochemical diversity is seen in prokaryotes, and you see bacteria are very simple and they carry on different biochemical activities, and moreover their identification is based on biochemical reactions that they carry on and they do. And these biochemical activities of bacteria enable them to produce a large number of products. As far as the shape and size of bacteria are concerned, bacteria may be circular or ring-shaped. These ring-shaped bacteria may be arranged in pairs regarded as the Diplococcus. They may be arranged in a chain regarded as Streptococcus. They may be arranged in groups of 4. They are regarded tetrads, T-E-T-R-A-D-S, tetrads. They may be arranged in a cuboidal form, like a cube, like a cubical box of chalk cube, say, and they are regarded as Sarcini. Sarcina is a genus and Sarcini, plural, the group Sarcini. So, these are the types of the cocci, and sometimes they may be spiral as Treponema palladium, which is shown here in the diagram. Treponema palladium, which is the causative agent of syphilis. Syphilis is a venereal disease. Sometimes the cocci may also be arranged as if in bunches of grapes, and these are regarded Staphylococcus. And Staphylococcus aureus is a very well-known bacterium. And sometimes they may be rods. The rods may be arranged linearly, one after one, as the cell divides, or they may be arranged one above another, as shown here. And while they are arranged one above another, the typical example is the Corynebacterium diphtheria, as if arranged as if matchstick are arranged in a matchbox. And because of this arrangement, a curtain-like structure is formed in the throat of the young children, and consequently children are unable to breathe and leading to the death of the child. And this is because of the arrangement of the cells of the Corynebacterium diphtheria, the causative agent of diphtheria. prokaryotes. Here, of course, the prokaryotes, blue-green algae or cyanobacteria comes under prokaryotes. Bacteria comes under prokaryotes. Archaeobacteria comes in the prokaryotes. Here, mainly I'm telling you regarding gram-positive bacteria and gram-negative bacteria. There is a stain which was discovered by Christian Gram, and this stain, on the basis of this stain, bacteria have been divided into two groups. One group is that they catch the stain, another group of the bacteria is that they do not catch the stain, Gram stain. The bacteria that which catch the stain, finally they appear purple to black after staining. And bacteria, they are regarded as the gram-positive bacteria. And on the other hand, gram-negative bacteria, they do not retain the gram stain. They appear the pink colored after staining with the differential staining, that is the safranin. So there are two broad categories of bacteria, gram-positive and gram-negative. So as far as their structure is concerned, gram-positive bacteria have got a wall outer to the cell membrane or plasma membrane. Here, this is the outer red colored one is the wall of the gram-positive bacteria, and inner is the wall. So this wall is because of a chemical substance known as the peptidoglycan. Peptidoglycan in turn is made up of chemical substances N-acetylglucosamine and N-acetylmuramic acid. N-acetylglucosamine and N-acetylmuramic acid arranged in a diagonal manner, and some bridges are also, peptides are there. In case of the gram-negative bacterium, which is here above, you see there are two membranes, outer membrane and inner membrane. Outer membrane and inner membrane is also regarded as the plasma membrane or cytoplasmic membrane. In between the two membranes, which are shown here with the blue color, is a space. This space is known as the periplasmic space. Periplasmic, periplasmic space. In between the periplasmic space, a red portion has been shown, a red line has been shown all around. This is known as the peptidoglycan. So, major difference between the gram-positive bacterium that is below and gram-negative bacterium that is above is that the layer of peptidoglycan is much thicker in the gram-positive bacterium in comparison to the gram-negative bacterium. This is 3 to 4 times thicker in case of gram-positive bacteria. As prokaryotes has got the naked genetic material, there is no membrane, and so this is the chromosome. The DNA is here. It is regarded the chromosome. Generally in the bacteria there is a single chromosome. Mostly the instances of two chromosomes are very rare, but generally there is a single chromosome, and there is another small ring. This ring is also made up of DNA and is regarded as the extra chromosomal ring of DNA, and this is known as the plasmid. This plasmid contains the gene and is capable of providing resistance to bacteria against drug, resistance to bacteria against heavy metals, and a number of characteristics are provided by the plasmid. So a flagellum is also there in the bacterium. This flagellum makes bacteria motile. motility to the bacterium is provided by this flagellum. Friends, if somebody asks you, what is the fastest animal in the world? Your reply will be tiger is the fastest animal because it moves 7 times of its body length in 1 second. But I may tell you, bacteria are much faster than the tiger itself. Bacterium can move 10 times of its body length in 1 second. So, bacteria are the fastest one. And besides, some fimbriae, some smaller appendages may be there, hair-like appendages may be there, which are known as the fimbriae. The function of the fimbriae is also attachment, and another appendage may also be there on the bacterial cell, not shown here in this diagram. That is the pilus. Singular is the pili. Pilus or pili, they help the bacterium in conjugation, and genetic material among the bacteria between two bacteria are shared through the pilus, and in some old books, in the old literature, here a sort of invagination of the membrane is shown, that is written as mesosome, mesosome. Mesosome has not been shown here in this diagram here, but this bacterial cell, and formerly it was believed that the mesosome helped in division of the bacterium, was presumed to help in the cell division of the bacterium, but here mesosome has not been shown, because now it has been proved that mesosome is formed as a result of faulty staining techniques and well-developed techniques nowadays that does not require the dehydration of the material in the electron microscopy. No mesosomes have been observed. So now it has been proved and concluded that mesosomes, they are artifacts and not the true structures. As far as the size of bacteria is concerned, some of them may be very large, some of them may be very 1 micron and even smaller than 1 micron. A cigar-shaped bacterium, that is Apelopicium and Phycelsonii, that is found in the gut of surgeonfish, is up to 600 micrometer or microns. So this bacterium, which is cigar shaped in structure, is 600 micrometer or microns in size. There is a term, tree of life. 3 primary branches of microorganisms have been derived. They are bacteria, rather also referred to as eubacteria, bacteria. Then archaea, that include the archaeobacteria, which are the oldest animals on the oldest bacteria or the oldest organisms on this world. If you have to find your ancestors, ancestry, and perhaps these are the oldest microorganisms. And then eukarya. Carl Woese is an American scientist who has derived these 3 on the basis of DNA, RNA groupings, also regarded as a DNA homology on the basis of, and archaea. The 3 groups in this way comprises the tree of life. The Didinium is a carnivorous protozoan of ciliates. It has got two layers of cilia or fringes, is about 150 micrometer in diameter, and it is engulfing and another protozoan. So how some cells might have been formed with the nucleus, and such organism, it is believed that one organism might have entered the another, and such organism exists today even. Two or dual organism exist today even, that provides the proof. And how nucleus might have been formed. Nucleated cells might have been formed from the prokaryotes. Perhaps a smaller prokaryote might have, the currents of water were there in the ocean where life created. Some of the smaller bacterium, some of the smaller prokaryote might have entered larger prokaryote. And in this way, this might have led to the formation of the first eukaryote. Two theories have been proposed in the formation of. One of them is the endosymbiosis. One microorganism, one prokaryote might have entered the larger prokaryote. And second is the theory of invagination. For example, the membrane might have, a part of the membrane might have invaginated, and gradually it might have got separated and of course remained inside the larger prokaryote, and in this way resulted the formation of the nucleated cell. It will be worthwhile to tell that there are no membrane bound tubules in the plasma membrane of prokaryotes other than cyanobacteria. Means in bacteria there are no membrane bound tubules except cytoplasmic membrane, but membrane bound tubules are present in cyanobacteria or blue-green algae that carry on photosynthesis, and these membrane bound tubules have been regarded as the thylakoids that contain the pigment for photosynthesis. And in the same way, origin of mitochondria might have taken place. A small bacterium might have entered a larger eukaryote, and while this bacterium entered, self-like structures might have been created as shown here, and this resulted into the origin of mitochondria. Mitochondria are also regarded as the powerhouses of the cell that help in creation of releasing energy. Not only mitochondria, eukaryotic cells has got some other structures also. These eukaryotic cells, the structures are, for example, in photosynthetic cells, there are chloroplasts. Of course, in prokaryote, thylakoids are there, but in photosynthetic cells, in eukaryote, in plant cell, one thylakoid, second thylakoid, third, in this way, membrane bound tubules or sacs, they are arranged one above another, referred to as grana, singular granum and plural grana, as if in a pattern, as if coins have been arranged one above another, a piles of coins, 10 coins and or 5 coins like that. So grana is like this, and other structures that may be there, Golgi bodies, they are the tubules, in the eukaryotic cells. Golgi bodies have been named as Golgi after the scientists who discovered them. These Golgi bodies, they have also been called dictyosomes. So these are the membrane bound tubules. There are certain vacuoles are there in the eukaryotic cells, contractile vacuole, that helps, they are particularly present in some protists and send out the waste material, and they also help the function of digestion. And so some other appendages may be there, flagella may be there on the some unicellular eukaryotic cells also as chlamydomonas, and there are some unicellular photosynthetic eukaryotic cells as chlorella that does not have the appendages. So variation is there, and mutations sometimes have taken place in the mitochondrial DNA and lead to many diseases including cancer, diabetes, heart disease, and age-related neurodegenerative disorder. So these are some of the diseases that have been reported recently in mutations that take place, sudden and abrupt changes in the DNA that are responsible for the mutation, that are responsible for the causation of certain diseases. And very, very recently, there has been some solution has been discovered for these mitochondrial diseases. Mitochondrial gene therapy has been solved by mitochondrial replacement therapy, abbreviated as MRT, that has been passed and approved by Human Fertilization and Embryo Authority, that is HFEA. So there are certain diseases because of mutation that result certain diseases, and solution to these diseases have been found, and even HFEA has permitted the solution in United Kingdom. Two methods have been proposed for the solution. The first one is the pronuclear transfer method. In the pronuclear transfer method, a mother's egg with abnormal mitochondria is taken and it is fertilized with a sperm. Side by side, two experiments are conducted. In one, mother's egg with abnormal mitochondria and a donated egg with normal mitochondria is also fertilized. So in the next step, a zygote is formed. In the first case, a zygote is formed with abnormal mitochondria, and in the second parallel case, zygote with the normal mitochondria. So the pronuclei removed from zygote with the abnormal mitochondria and then transferred to the empty cell, pronuclei removed, the pronuclei of which have been removed, and then embryo with normal mitochondria is placed into the uterus for further development. So you see here, this is with here AM stands for abnormal mitochondria and NM stands for normal mitochondria. The below is the donated egg. This is from the mother and sperm from the father, and then this in the next stage it is with abnormal. A stands for abnormal, M stands for mitochondria, and here normal mitochondria. And these nuclei they are removed and are passed on. Here they have been removed, and then these two pronuclei are passed on to it, and consequently here it has been passed into the egg, the pronuclei of which have been removed and deleted, and further it forms a fused-celled embryo which is transplanted into uterus of the mother. Here a drawback of this is that this method is that two zygotes have to be destroyed. This zygote and this zygote. So this is the drawback of the method, and there is an alternative method, that is the spindle transfer method, that is also there. In the spindle transfer method, unfertilized mother's egg with abnormal mitochondria is taken, and unfertilized donated egg with normal mitochondria are taken. Then spindle and associated chromosome are removed from the mother's egg and transferred to the empty donor egg. And in the lower case, the spindle and associated chromosome removed from donated egg and discarded. And of course, both for the unfertilized one, and the spindle is transferred to the donated egg, and then that is fertilized with a sperm of father and embryo with normal mitochondria, and maternal and paternal genome are transferred to the uterus. So this in the former method two zygotes were destroyed, but in this initially two eggs are taken, with one with the MA and one with the normal and abnormal mitochondria. So this is how the rectification of the diseases are done, and a number of diseases like cancer, diabetes, heart attacks and other diseases are avoided in this way. Recently, another problem has been created, that is the bacteria resistant to last resort antibiotic, that is the carbapenems, discovered in the swine farming. Scientists of Ohio State University have identified that certain bacteria have got resistant, that were isolated from the swine fecal matter, and these bacteria, they have got resistance against the carbapenem, which is one of the most powerful antibiotic. So imagine what will happen. In continuation, this carbapenem, they harbor beta-lactamase gene IMP27 that make them resistant to carbapenem antibiotics. So this particular gene provides them provide them power to resistance, and this perhaps is because of the excessive use of antibiotics for which Louis Pasteur had warned us. Bacteria resistant to carbapenems are there 18 isolates of the Enterobacteriaceae, which are pathogens like Escherichia coli, a common bacterium, and Klebsiella pneumoniae. They are included in it. They have got resistance, exhibit resistance, and the structure of the carbapenem, they have got R1 group, R2 group, R3 group, and COOH group, and O with double bond. And this is the structure of carbapenem. So this was a powerful antibiotic, but certain bacteria have been created. They have gained resistance against this also, and now time has come to discover some newer antibiotics. Carbapenems are antibiotics used for treatment of infections known as suspected to be caused by multi-drug resistant MDR bacteria. Their use is primarily in people who are hospitalized. So this is how carbapenems have failed. In continuation, carbapenems like penicillins and cephalosporins are members of beta-lactam class of antibiotics which kill bacteria by binding to penicillin, binding proteins, and inhibiting cell wall synthesis. They exhibit a broader spectrum of activity compared to cephalosporins and penicillins. So, how they have created resistance? Because of the beta-lactam rings. Carbapenems are effective in effectiveness is less affected by many common mechanisms of antibiotic resistance in comparison to other beta-lactams. Of course, it is less in comparison to other beta-lactams. The carbapenems antibiotics were originally developed at Merck and Company from the carbapenem phenamycin, a naturally derived product of Streptomyces cattleya. Streptomyces cattleya, how it has been given name. Cattleya is an American orchid, and a fascinating nomenclature. And Streptomyces, you know, they are a genus of bacteria. Carbapenems concern has arisen recently over increasing rates of resistance to carbapenems, as there are few therapeutic options for treating infections caused by carbapenem resistant Enterobacteriaceae, including Klebsiella pneumoniae. Klebsiella pneumoniae is a pathogen. Friend, a very interesting question comes that the why penicillin that kills the bacteria but does not harm our blood cells. The simple answer is the bacterial cells has got peptidoglycan, and our blood cells, they do not have the peptidoglycan. That's why penicillin does not affect the blood cells, and so some other antibiotics also. Friends, some more information you can learn from my text. I have authored 2 texts. Introductory Microbiology. You can find more information, more diagrams, and more illustrations, and fascinating questions at the end of each chapter. And another book, Microbiology Questions and Answers. These are my 2 most popular books. The Microbiology Question and Answer, very fascinating, interesting questions have been answered in a very lucid style, and you'll find many original ideas in it. Thank you. Friends, remain in contact with for my next lecture. Thank you.